Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about the synthesis of fatty acids. In previous videos, we talked about the three metabolic states of a human being: postprandial, which is after a meal, early starvation or fasting, which is between meals, and if you have not eaten for one day, then we get into late starvation. So in this video, we shall continue with the discussion on this state here, the postprandial state. In the last video, we talked about how glucose that we get from our diet after a meal, some of this glucose will be used to replenish the glycogen in the liver that was used for maintaining blood glucose during starvation periods. So besides glycogen, there's another very important fuel. There's another very important fuel that is necessary for energy generation, and that is fatty acids. So today we'll talk about how we synthesize fatty acids after a meal. Fatty acids are mostly made in the liver as well as the adipocytes. So adipocytes and the liver are the two major places in the body where we make quite a bit of fatty acids. So let's look at the key steps of fatty acid synthesis. Now, fatty acid synthesis is a cytoplasmic pathway, so it occurs in the cytoplasm, and it requires two very important things. It needs the raw material, the basic building block, and that building block is acetyl-CoA. It requires a lot of acetyl-CoA, and then it requires this coenzyme called NADPH. As we mentioned, this synthesis occurs predominantly in the liver and the adipose tissue. The NADPH comes mostly from a pathway called the hexose monophosphate shunt. We'll talk about this pathway in a future video. So cells that make lipids, that synthesize lipids, this pathway, HMS pathway, is a very important pathway for these cells, such as the hepatocytes in the liver as well as the adipocytes of your adipose tissue. The acetyl-CoA, that can come from glucose. We saw how in Main Street acetyl-CoA can be derived from glucose, or it can come from other sources, including from breakdown of fatty acids as well as from some amino acids. Now, since you need acetyl-CoA to make fatty acid, and unfortunately all the acetyl-CoA or most of our acetyl-CoA is not in the cytoplasm where fatty synthesis occurs. Most of our acetyl-CoA is in the mitochondria. So the first thing the cell would have to do is to send this acetyl-CoA out from the mitochondria into the cytoplasm. So how is this done? So this is done by transporting citrate. So the acetyl-CoA, if you remember from Krebs cycle, acetyl-CoA and oxaloacetate can combine together to form citrate. So this citric acid or citrate is transported out of the mitochondria by this tricarboxylate transport protein, and that citrate comes out into the cytoplasm. Now in the cytoplasm, the enzyme ATP citrate lyase will break down this citrate into oxaloacetate and acetyl-CoA. So now we have this acetyl-CoA in the cytoplasm, and this acetyl-CoA can be used to make the fatty acids. This oxaloacetate will be reconverted back to pyruvate, and that will be transported back into the mitochondria. So this is how we bring the acetyl-CoA from the mitochondria into the cytoplasm, where fatty acid synthesis will occur. So now let's look at what happens to this acetyl-CoA, how is it made into fatty acids. So in the first step of fatty acid synthesis, the acetyl-CoA that's now in the cytoplasm is converted into malonyl-CoA. So basically this is a reaction where carbon dioxide is joins right here, this carbon. So you see the carbon dioxide joined to the acetyl, to this acetyl group, to form what we call malonyl-CoA. So because this is joining carbon dioxide, we call this carboxylation reaction. Okay, so we're taking carbon dioxide, reacting it with a substrate, and we are forming a carboxyl acid. So this is a carboxylic acid. This is called a carboxylation reaction. The enzyme that catalyzes this reaction is called acetyl-CoA carboxylase. So carboxylases are enzymes that catalyze carboxylation reactions. Carboxylase enzymes belong to the family of enzymes called ligases. Okay, so carboxylases belong to this family called ligase. This enzyme acetyl-CoA carboxylase requires the vitamin B7. So vitamin B7, which is also called biotin. So in order for this reaction to happen, the enzyme requires biotin to catalyze this reaction. Acetyl-CoA carboxylase is the most important enzyme that regulates fatty acid synthesis. So this very important enzyme, acetyl-CoA carboxylase, or ACC, that regulates fatty acid synthesis, this enzyme can exist as its unphosphorylated form, doesn't have a phosphate, that's the active form, or it can exist in its phosphorylated form, which is the inactive form. The phosphorylation of ACC, where ACC reacts with ATP to give you the phosphorylated enzyme in ADP, this reaction is catalyzed by the enzyme called AMP-dependent protein kinase, or AMPK. Insulin stimulates this enzyme here, the dephosphorylated form. Okay, so insulin stimulates dephosphorylation of this enzyme to give you the ACC active form. And when ACC is active, we will see this first step in fatty acid synthesis happening. So insulin not only stimulates glycogen synthesis after a meal, insulin also stimulates fatty acid synthesis after a meal, because insulin is only high, the levels of insulin is high only after a meal. Okay, so the cell now has, it has taken some acetyl-CoA and made into malonyl-CoA. So now the cytoplasm has acetyl-CoA as well as malonyl-CoA. So now we are ready for the rest of the synthesis, and the rest of the synthesis, fatty acid synthesis, is carried out by this very big enzyme called fatty acid synthase. Fatty acid synthase has a shape that looks like an X. It has two halves, so you can see basically this is one half of the enzyme and this is the other half. So it's basically a sort of a dimer. Each half is made up of a protein, a cofactor called ACP. ACP stands for acyl carrier protein. This is a small protein with a 4-phosphopentanine prostatic group, very similar to what you see in coenzyme A. Then each half also has 6 active sites. Okay, 6 active sites. 1, 2, 3, 4, 5, 6. So each half of the enzyme has these 6 catalytic sites, which you will see it will catalyze 1, 2, 3, 4, 5, 6 reactions. So let's look at the steps by which fatty acid synthase synthesizes the fatty acids. So in the first reaction, the acetyl-CoA reacts with the ACP group here, and the acetyl group basically is transferred to the ACP. So the acetyl group will be connected to the ACP. So this reaction between acetyl-CoA and the ACP is catalyzed by that catalytic site called MAT, which is malonyl acetyl transacylase. So this MAT catalytic site will catalyze this first reaction here. In the next step, the acetyl group, which is connected to the ACP part of the protein, of the enzyme, is transferred to this enzyme, Ks, is transferred to this beta-ketoacyl synthase enzyme right here. So the acetyl group that was here will be transferred now to this Ks. So the acetyl group will now be here and no more on ACP. So this is the next reaction that happens. In this case, the ACP becomes free while the enzyme, the synthase, has an acetyl group connected to it. So this acetyl carrier protein that is free now will pick up a malonyl. So it will react with malonyl-CoA, and the malonyl part will be connected to the ACP. So the ACP now carries a malonyl. Meanwhile, remember, there is an acetyl attached here. This reaction is also catalyzed, just like before, by the MAT catalytic site. In the next reaction, the acetyl group, this group that is attached to the Ks catalytic site, is transferred to the malonyl group. So you can see this carbon here of malonyl reacts right here, and we get this 4 carbon, 1, 2, 3, 4 carbon intermediate that is attached to the ACP arm. This reaction is catalyzed by the synthase, and the carboxyl part of the malonyl comes out as the gas carbon dioxide. So remember, the carbon dioxide in the first reaction that was catalyzed by ACC, the carbon dioxide became the carboxylic part of malonyl. However, the acetyl, this acetyl really comes from acetyl-CoA. So in a way, this is really the acetyl carbons of malonyl-CoA. So this means that in this intermediate here, this 4 carbon intermediate that is connected to the ACP part of the enzyme, all these 4 carbons really have come from acetyl. Okay, because the only carbon that did not come from acetyl was the carboxyl group, but that was eliminated as carbon dioxide. That came from carbon dioxide, and it is now leaves as carbon dioxide. Okay, so now this ACP has your 4 carbon intermediate, this 4 carbon intermediate attached to the ACP part of the fatty acid synthase. So in the next step, the enzyme K, the catalytic site KR is beta-ketoacyl reductase. So that's a reductase. So this enzyme catalyzes the reduction of this carbon here from a C double bond O to a hydroxyl group. And you can see for this reaction to happen, we need the reducing agent NADPH, which, as mentioned earlier, most of this comes from HMS. Now, it should be noted that as citrate is transported from the mitochondria to the cytoplasm, there is another reaction catalyzed by malic enzyme, and that also produces NADPH. So some of the NADPH for fatty acid synthesis can also come from this enzyme, malic enzyme reaction, besides the HMS. Okay, so besides HMS, this is also another source of NADPH in the cytoplasm. In the next reaction, water is eliminated. So basically this OH and one of the hydrogens is eliminated, and we get a double bond form. This reaction is catalyzed at the catalytic site DH. DH is beta hydroxyl dehydratase. And in the last step of this cycle, the double bond is reduced to a single. Again, NADPH is required, and this part is catalyzed by the catalytic site ER, and ER is enol reductase, so that's a reductase enzyme. So at the end of this cycle, we now have 4 carbons. All 4 carbons really came from acetyl, because remember the malonyl carboxyl group comes out as carbon dioxide. So now we have these 4 carbons that are attached to the ACP part of the enzyme. Now this intermediate here now will simply be repeated. This cycle will be repeated. So instead of putting this here, let's just put this 4 carbon intermediate here and then repeat the reactions. And if you do that, the 4 carbons will then become 6 carbons and 8 carbons and 10 carbons, all the while attached to this ACP part of the protein. Okay, so the fatty acid starts growing 2 carbons at a time, gets longer and longer. After 7 cycles, we will get this 16 carbon fatty acid, palmitoyl, that is connected to ACP. The catalytic site thioesterase, TE, then cleaves the ACP part from the palmitic acid, and that will give us palmitate, and the ACP will come back to its original form. So the major fatty acid that is derived from the fatty acid synthase is this C16 palmitic acid. There is also a little bit of C18 and C14 fatty acid that may be formed during these reactions. So it's important to note that all of the carbons of these fatty acids that we synthesize, they all are derived from acetyl-CoA. So once a fatty acid is made, we can increase the length of the fatty acid chain. We can elongate the fatty acid. And this is done by 2 processes. The major pathway is the one that happens in the endoplasmic reticulum, the microsomal part, endoplasmic reticulum. This is the major pathway of how we humans elongate our fatty acids. There is also a minor pathway that occurs in the mitochondria. So let's look at this major pathway that occurs in the endoplasmic reticulum first. So the fatty acid will react with the malonyl-CoA. So the 2 carbons of the malonyl-CoA will be donated to the fatty acid. The carboxyl group as before, comes out as carbon dioxide, and the coenzyme A here also comes out. And this reaction is catalyzed by a synthase enzyme. This enzyme is also called elongase. So this 3-keto-acyl synthase is also called elongase, or sometimes abbreviated as ELOVL, which stands for elongation of very long chain fatty acids. In the next step, this carbon here is reduced using NADPase as a reducing agent. It is reduced to an alcohol, and then water is removed to form a double bond. This is catalyzed by the dehydratase enzyme. And finally, the double bond is reduced to a single using NADPase as a reducing agent, and this again is carried out by a reductase enzyme. So in this way, the fatty acid, the original fatty acid, is lengthened by 2 carbons. Okay, so at every time the fatty acid increases its chain length by 2 carbons, and these 2 carbons really come from acetyl-CoA. And why acetyl-CoA? Because remember, the malonyl-CoA carbons that you find here are actually derived from acetyl-CoA. Okay, so the only carbon, the carboxyl carbon, never appears in the fatty acid. It always comes out as carbon dioxide. The mitochondrial pathway is very similar to this previous pathway we talked about. And this is also very similar to beta oxidation. This is how fatty acids are broken down. And we'll talk about this very important beta oxidation under starvation. This is how we break down fatty acids to get energy. So the minor pathway of elongation happens in the mitochondria, and you can see basically the same things are happening here too. You can see in this case, instead of malonyl-CoA, we are using acetyl-CoA. So the acetyl-CoA carbons are being donated here to the fatty acid, and then there is the reduction using NADH, removal of water, elimination of water, forming a double bond, and then the double bond is made to a single bond by another reduction where NADPase is used to give us the elongated fatty acid. So for example, if we started with a 16-carbon fatty acid here, palmitic acid, then we end up with a 18-carbon fatty acid, stearic acid. Okay, so this is how we can make fatty acids longer, and then this fatty acid can further be elongated to a 20-carbon and 22-carbon and so on. Now, besides elongating fatty acids, we can also form unsaturated fatty acids. So we can desaturate the saturated fatty acids. We have enzymes such as the delta 9 or the delta 6 or the delta 5 or the delta 4 fatty acyl-CoA desaturases. Okay, so these desaturases can add double bonds to the fatty acids. So for example, if we look at this delta 9 fatty acyl-CoA desaturase, it will add a double bond between 9 and 10, so between carbons 9 and 10. So for example, stearoyl-CoA will become olyl-CoA, so stearic acid is basically converted to oleic acid. And these enzymes require NADH as well as molecular oxygen. So the desaturase enzymes that we have, they cannot add double bonds beyond position 9. So that means we require 2 very important fatty acids in our diet. These are called essential fatty acids, and they are linoleic acid. You can see linoleic acid has a double bond at position 12. Okay, so we do need linoleic acid in our diet. And we also need alpha-linolenic acid, which has again a double bond at position 12 and 15. Okay, so we cannot make double bonds at those positions more than 9. So we also need ALA, or alpha-linolenic acid, in our diet. Okay, so these are the major points for fatty acid synthesis, fatty acid elongation, and fatty acid desaturation. We shall talk about other pathways of lipid synthesis in a future video. As always, thank you very much for watching. Goodbye and have a very happy and a healthy day.